Hello everyone and welcome back to Dr. Wasif Academy. This is the place where we bring you authentic, high quality and updated educational content to help you master your subjects with confidence. Today, we are diving into one of the most important and fascinating topics in biology, that is enzymes. In this lecture, we are going to cover what is an enzyme, how it works, when it works, and where it works. So grab your notebooks and let's get started. Before we look at how enzymes work, we first need to understand what they actually are. We often hear about speeding up life. Well, this is exactly where it happens. First of all, let's talk about the word catalyst. Simply, a catalyst is any substance which increases the rate of a chemical reaction. Think of it like a shortcut on a map. It gets you to the destination much faster. So crucially, a catalyst is not used up. Remember it, catalyst is not used up in any reaction. It remains chemically unchanged once the reaction is over. Okay, so this leads us to our first key concept. A little goes a long way because the catalyst is not destroyed in any chemical reaction. It can be used over and over again. Now, what is an enzyme? Simply, an enzyme is a biological catalyst. Remember this word, biological catalyst. It will be very helpful in your exam. So, almost every enzyme you will study is a protein. It means all enzymes are protein in nature. Their job is to speed up metabolic reactions, which are just the chemical reactions happening inside a living organism. So, without enzymes, these reactions would happen so slowly that life as we know it's simply it could not exist. Now a quick tip for your exam for preparation. You must remember that keywords are everything. Keywords are play very critical role in your exam. So when an examiner asks for a definition of enzyme, you must use two specific terms. First one is biological catalyst and second one is protein. Do not just say that enzymes speed up things. Okay, use these technical words, this technical terminology to show you truly understand the science. As we have defined enzymes as biological catalyst, but why are they actually so important? To put it simply, without them means without enzymes, the chemistry of life would move at a snail's pace, means very slow. Enzymes allow crucial chemical reactions to happen fast enough to keep us alive. This is especially impressive because they do this at relatively low temperatures like our normal body temperature, that is 37 degrees Celsius. So without enzymes, we would need much higher temperatures to trigger these reactions, which would unfortunately be fatal to our cells. You can think of enzymes as the master controller of metabolism. Metabolism is just a fancy word for the sum of all the chemical reactions happening inside our body. So enzymes ensure these reactions happen in the right order and at exactly the right speed. Let's look at three big examples where enzymes are the stars of the show. First of all is digestion. Enzymes break down large insoluble food like starch into tiny soluble molecules like glucose, so your body can absorb them. Think of examples are amylase or lipase. The second one is the process of respiration. This is how your cells release energy. It is a complex chain of events and every single step is guided by a specific enzyme. The thirdly, that is also very important process, a very important metabolic process in our body, that is DNA replication. When your body makes new cells, enzymes like DNA polymerase help unwind your DNA and build new strands with incredible accuracy. Like you can see in this picture, this is you can say is DNA polymerase. Okay, So when it binds, it is helpful in DNA replication. It speeds up the reaction. To really understand their importance, means the importance of enzymes, let's look at what if scenario. If you suddenly had no enzymes in your body, what will happen? These vital metabolic reactions would not just be slow, they would be so incredibly sluggish that your body could not function. An organism would literally die before a single meal could be digested or enough energy could be released. So they are not, these enzymes are not just helpful, these are essential for survival. And you can see both scenarios in this picture. Okay, this is scenario A, that is life without enzymes, and this is scenario B, that is life with enzymes. Here you can actually understand that what happens if we have no enzyme in our body. Okay. Now let's talk strategy for your exams. To get top tier marks in your exams, you should understand it simply what how we should respond to the examiner's questions. The examiners do not just want to hear that enzymes are used in digestion; they want precision. So instead of a general statement, try saying, in the mouth, the enzyme amylase breaks down starch into maltose. Similarly, try to link the enzyme to its location. For example, mention that respiration enzymes work in the mitochondria, or that specific digestive enzymes are produced in the pancreas. So, being precise shows the examiner that you truly understand the biological system. My dear students, did you ever think that why your body needs hundreds of different enzymes instead of just one giant super enzyme which can perform all the functions? Okay, the answer lies in one word, that is specificity. So what does specific mean? What does that mean? In biology, being specific means an enzyme is highly selective. One type of enzyme will typically only catalyze one specific chemical reaction. It focuses its energy on one particular substrate, which is the molecule the enzyme acts upon. What is substrate? Substrate is the molecule that enzyme acts upon. We shall discuss it later in this lecture. That will be in detail. So if we discuss about the cause, means a precision fit, the question is why does this happen? Means why a specificity is necessary. It is all about the 3D shape. Every enzyme has a very special area called the active site. Think of the active site like a specialized docking station. Okay, specialized docking station. For a reaction to happen, the substrate must have a complementary shape. Remember this word, complementary shape. Complementary shape is that which matches the active site perfectly. It is the famous lock and key model. Okay, so just as only one specific key can open your front door or your drawing room or your bedroom, so likewise only one specific substrate can fit into an enzyme's active site. For example, let's look at two examples which you will see often. The first one is about amylase. Okay, in this picture you can visualize the lock and key model and the how amylase performs its function. So if we talk about amylase, its active site is a perfect match for starch. Mean amylase, what does it do? It helps in the digestion of the starch. So it breaks down starch down into maltose, but it completely ignores protein. You can see here that it is effective for amylase is effective for starch, but it is not effective for protein because it does not fit there. So it completely ignores protein because they simply do not fit. The second one is about, the second example is about protease. This enzyme is built for proteins. Protease is built for proteins. Its active site fits peptide bonds perfectly, but it cannot bind to or break down starch. This is about specificity. That specific enzyme can act with specific substrate. This is complementary. Okay. So here is a golden rule for your exams. This one is the golden rule for your exam. If you write the substrate and enzyme are the same shape in your answer, you will likely lose the mark. Instead, always use the word complementary. They are not identical. What does complementary mean? Enzymes and substrate shapes are not identical. They are matching opposites which fit together. So using this one word shows the examiner you have a high level understanding of molecular biology. As we have talked about the lock and key, but let's take a closer look at the lock itself. This brings us to the most important part of the enzyme, that is the active site. So what exactly is active site? The active site is a specific region, usually a small pocket or a cleft on the surface of the enzyme. It is not just a random spot. It is the precise location where the substrate binds to the enzyme to start the reaction. So we can say structure determines function. As we have discussed earlier, structure determines function in biology. The active site has a very specific 3D shape. Because this shape is complementary to the substrate, they fit together perfectly. If that shape changes even a little bit, the key will no longer fit and the enzyme would not work. So how it works? When the substrate docks into the active site, they form what we call an enzyme-substrate complex. You can see it here that this is lock and key model, okay? Active site and enzyme complementary substrate key, and this is the enzyme substrate complex, ES complex, okay? This is enzyme substrate complex. So these both shapes would be complementary to each other. This is the temporary union, okay? This temporary union is where the magic happens. It loads the energy needed for the reaction, okay, and it allows everything to happen at high speed. Remember, enzymes are proteins in nature, as we have discussed in earlier slides, okay. So that means the shape of the active site is decided by a long chain of amino acids. The way this chain holds and twists creates the unique pocket, okay. So if the folding is wrong, the active site is wrong. This exam tip is for those who are looking for higher marks. You should understand that precision is very important. This is vital. When defining the active site, do not just say it is where the reaction happens. That is a bit too vague statement. Okay. Instead, use the phrase the region. What is active site? Active site is the region where the substrate binds. Also, always emphasize that the active site has a specific shape which is complementary to the substrate. So, including these 3 points, that is the region where it binds and the specific shape, and the third point is complementary. So, if you include these 3 points in your statement, in your answer, binding, specific shape, and complementary, this is the golden ticket to a perfect score. Now, to make sense of all these technical terms, scientists often use one of the most famous analogies in biology, that is the lock and key model. Think about your front door of your house. You have a specific lock, that is our enzyme, okay? And inside that lock is a unique internal mechanism, that is the active site, okay? The mechanism is the internal mechanism, that is the active site. Now you might have 10 different keys in your pocket, but only one has the exact right ridges and grooves to fit inside and turn that lock open. That winning key is our substrate. So what is the science, you can say, behind science, okay? While lock and key is a simple way to remember it, the scientific reality is all about complementary shape. The 3D structure of the enzyme's active site perfectly mirrors the 3D shape of its substrate. As you can see in this picture, this is a perfect visual explanation of this lock and key model. This perfect fit is what allows the chemical reaction to happen so efficiently and at a speed of light. So if a molecule does not match even by a tiny bit, it simply cannot bind. This is how your body maintains total control over its metabolic reactions and metabolic pathways. So it ensures only the right reactions happen at the right time at the right place. I want you warn here about a common trap in exams. Avoid, again I say, again I am stressing, again I am warning you and stressing that avoid the same shape trap. I see many of students write that the enzyme and substrate have the same shape. So if you do this, you will likely lose marks. Why? Because these shapes are opposite. Okay, one is a pocket, means the cleft, and the other fits into that pocket. So these are different shapes. So in your exam, always use the term, I'm again saying, always use the term complementary shape. So if you find that word hard to remember, you can say they fit like a lock and key. But complementary is the gold standard, okay, which examiners love to see. Now that as we have mastered the lock and key theory, we have understood that, let's see the actual four-step process of an enzyme in action. Now let's put everything together and see the life cycle of an enzyme controlled reaction. This happens in four clear logical steps. Step 1 is approach. The substrate molecule is moving around by diffusion until it eventually drifts toward the enzyme molecule. Second step is binding. The substrate collides with the active site and docks into place. Because their shapes are complementary, they fit together perfectly. This creates a temporary but vital structure called the enzyme-substrate complex. This is the moment where the actual work begins. Then, the step 3 is conversion. While both are enzyme and substrate are joined, the active site may adjust its shape slightly, and that process is known as induced fit to lower the activation energy. This allows the chemical reaction to happen and the substrate is transformed into our product. The step 4 is release and reuse. Because the products now have a different shape than the original substrate, they are released from the active site. But look at the enzyme, it remains completely unchanged. Its active site is now empty and ready to grab a new substrate and do this process all over again. Okay, in this picture you can visualize this approach, binding, conversion, and the fourth step, that is release and reuse. So when you are answering a question about enzyme action, use a numbered sequence just like we discussed here. This shows the examiner you understand the logical flow. Secondly, do not forget to use a term, that is enzyme-substrate complex. This is often a required keyword for getting higher marks. And finally, always mention that the enzyme is reusable. That is the fundamental definition of a catalyst, that it is unchanged. As we have studied the perfect scenario of enzyme-substrate complex, but the question arises, what happens when the environment changes, means temperature changes, for example, or the pH changes, what happens then? In our next slide, we will discuss how temperature and pH can affect these hard-working proteins, which are enzymes. Okay, dear students, that wraps up today's session. We have covered the fundamentals of the enzyme substrate complex, the lock and key model, and the mechanics behind enzyme specificity. If you found this lecture valuable, please subscribe and share it with your peers. It really helps the channel more than you know. And if any part of today's lesson was unclear, drop a comment below and I will get back to you. Thank you for watching and I'll see you in the next video. Till then, I wish you all best of luck. Goodbye.